Hi, my name is Emma.
And my name is Miranda.
Today we are going to teach you all about mitosis.
Mitosis is when a cell divides and makes two identical daughter cells. Mitosis can occur in a cell through these stages: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. The first phase in mitosis is interphase.
In interphase, there are three important checkpoints. The first checkpoint is the G1 checkpoint. The G1 checkpoint checks for
growth, metabolic activity, centriole replication.
If these fail, the cell goes into G0 phase, meaning it does not continue into mitosis.
The next part is called S phase. This is where the DNA synthesis occurs.
Lastly, G2 phase checkpoint is the final checkpoint. It prepares the cell for division.
But before we move on, it's important to know that interphase is where the cell spends most of its time, and chromosomes cannot yet be distinguished in the nucleus. Now we have prophase.
There's early prophase and there's late prophase, also known as prometaphase.
In early prophase, centrioles begin to move away from each other. Chromosomes consisting of two sister chromatids are now visible inside the nuclear envelope. The nuclear envelope/membrane starts to disappear.
Late prophase, or known as prometaphase, is where the nuclear envelope is fully dispersed in the cell. The spindle apparatus begins to form, and that completes prophase.
Now we have metaphase.
The attached sister chromatids begin to move into the middle or metaphase plate.
It's like a tug-of-war between the spindle fibers until the sister chromatids separate at the centromere.
And that completes metaphase.
Next is anaphase.
Anaphase represents the movement of sister chromatids in opposite directions away from the middle or metaphase plate. At the end of anaphase, the cell starts to separate.
This is the shortest phase in mitosis, and that completes anaphase. Lastly is telophase and cytokinesis. This is where you can distinctively see the cleavage furrow. A cleavage furrow is the pinching of the parent cell until it separates into two cells.
This only happens in animal cells. Plant cells would rather form a cell plate which extends from the middle or metaphase plate outwards across the cell. The chromosomes uncoil and then become indistinct.
A nuclear membrane begins to form around the chromosome clusters.
The spindle apparatus begins to break down.
And now we have generated two identical new daughter cells that now can begin mitosis.
Remember, only animal cells and plant cells can go through mitosis.
And that completes mitosis. Thanks for watching! Now that we have learned about each phase, let's do a little review. First, let's start with interphase.
Interphase has the G1 phase, the S phase, and the G2 phase.
In the G1 phase, there's rapid growth and metabolic activity and centriole replication.
At the G1 checkpoint, if the cell has not completed those requirements, it does not further the process.
In the S phase, there's chromosome replication and DNA synthesis.
At the G2 phase, growth and final preparations for cell division happens.
Remember that this is where the cell spends most of its time. Next we have is prophase. Remember, there's early prophase and late prophase, also called as prometaphase.
In early prophase, centrioles begin to move away from each other, and two sister chromatids are now visible. The nuclear envelope also starts to disappear.
Okay, now in late prophase, or known as prometaphase, the nuclear envelope is fully dispersed, and the spindle apparatuses begin to form. Now that completes prophase. Right. Now metaphase.
Sister chromatids move into the middle or metaphase plate.
The spindle fibers tug on each other until the sister chromatids separate at the centromere. And that is the end of metaphase. Anaphase. In anaphase, there's movement of sister chromatids away from the metaphase plate.
This is the shortest phase in mitosis.
At the end of anaphase, the cell starts to separate. And now let's do a little review on the last two phases, telophase and cytokinesis.
You can distinctly see the cleavage furrow, And remember, this only happens in the animal cell.
The chromosomes will uncoil.
The nuclear membrane will form around the uncoiled chromosomes.
And the spindle apparatus breaks down. Now that we have created two identical daughter cells, we will show you how they divide next.
So, the daughter cells can both go through cell division again and create two more identical daughter cells, and then further the process. And all of these are identical, which help form tissues in the body.
Okay, so this explains how cells divide and reproduce. And this is the end of our mitosis review. Thanks for watching!